Unit 3, Cell Structure and Functions. In that, the chapter 8, Cell, the Unit of Life. Let's start reading the chapter. Biology is the study of living organisms. The detailed description of their form and appearance only brought out their diversity. It is a cell theory that emphasizes the unity underlying this diversity of forms, that is, cellular organization of all life forms. A description of cell structure, cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around the living phenomena, that is, physiological and behavioral processes. This mystery was a requirement of the integrity of the cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take a physico-chemical approach and use cell-free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analysis of living tissues for elements and compounds. It will tell us what types of organic compounds are present in living organisms. In the next stage, one can ask the question, what are these compounds doing inside a cell? And in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes? It can also explain the abnormal processes that occur during any diseased condition. This physico-chemical approach to study and understand living organisms is called reductionist biology. This is called the reductionist biology. Okay, so let me underline reductionist biology. The concepts and techniques of the physics and chemistry are applied to the and applied to understand biology. In chapter 9 of this unit, a brief description of the biomolecules is provided. Moving on, need to know the scientist G.N. Ramachandran, an outstanding figure in the field of protein structure, was the founder of the Madras School of Conformation and Analysis of Biopolymers. His discovery of the triple helical structure of the collagen published in Nature in 1954 and his analysis of the allowed conformations of the proteins through the use of the Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on October 8, 1922 in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had a considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as a top ranking student in the BSc Honors Physics course of the University of Madras. He received a PhD from Cambridge University in 1949. When at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications on the models of the alpha helix and beta sheet structures. He directed his attention to solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. G.N. Ramachandran, 1922 to 2001. Let's go into the topic then. The chapter 8, the cell, the unit of life. Cell, the unit of life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. They are performing the essential functions of life. Wait a minute. So, unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of cell does not ensure the independent living. Hence, the cell is a fundamental structural and functional unit of all living organisms. The Anthony von Leeuwenhoek, the first scientist described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Moving on, cell theory. Cell theory. In 1838, in 1838, the Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Schwann, 1839, a British zoologist, studied the different types of animal cells and reported that the cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is unique character of the plant cell. Presence of cell wall is a unique character of the plant cell. On the basis of this, Schwann proposed the hypothesis. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Sleeden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed, but Rudolf Virchow first explained that the cell divided and new cells are formed from pre-existing cells, like what he said, omnis cellula e cellula. Rudolf Virchow said, omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and products of cells. All cells arise from pre existing cells. An overview of cell. You have earlier observed the cells in an onion peeler and our human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, the onion cell is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is a cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material, DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas the cells that lack a membrane bound nucleus are prokaryotic. Eukaryotic and prokaryotic. Need to see the difference between the eukaryotic and prokaryotic. Cells that have membrane bound nuclei are called eukaryotic, whereas cells that lack a membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living stage. Besides the nucleus, the eukaryotic cells have Besides the nucleus, the eukaryotic cells have other membrane bound distinct structures called organelles, like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane bound organelles. Prokaryotic cells lack the membrane bound organelles. Such membrane bound organelles are absent in prokaryotic cells. Ribosomes are the Non-membrane bound organelles found in all cells. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Then within the cell, ribosomes are found not only in the cytoplasm, but also within the two organelles, chloroplast in plants and mitochondria and on rough endoplasmic reticulum. The ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast and mitochondria, and in rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centriole, which helps in cell division. Animal cells contain another non-membrane bound organelle called centriole, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, the smallest cells, we can say, are only zero point 0.3 millimicron in length, while bacteria could be 3 to 5 millimicron. The largest isolated single cell is the egg of an ostrich. The largest isolated single cell is the egg of an ostrich. Among the multicellular organisms, the human red blood cells are about 7.0 millimicron in diameter. The nerve cells are some of the longest cells. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like or polygonal or columnar or cuboid or thread-like or even irregular. The shape of the cell may vary with the function they perform. Let's take a look on the prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pneumonia-like organisms. They're generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of the bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma shape, and spirillum. The organization of the prokaryotic cell is fundamentally similar, even though even though prokaryotes exhibit a wide variety of shapes and function. The organization of the prokaryotic cells is fundamentally similar. All prokaryotes have a cell wall. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. Have an exception, that is mycoplasma. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The field matrix filling the cell is the cytoplasm. The fluid matrix filling the cell is cytoplasm. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA we call genomic DNA, many bacteria have small circular DNA outside the genomic DNA. That many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibodies. Such character is resistance to antibiotics. Here, not antibodies, antibiotics. One such character is resistance to the antibiotics. In higher class, you learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cell except for ribosomes. No organelles, see this? No organelles like ones in eukaryotes are found in the prokaryotic cell except for ribosomes. The prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is a characteristic of prokaryote. A specialized differentiated form of cell membrane called mesosome is a characteristic of prokaryote. They are essentially infoldings of cell membrane. Mesosomes are essentially infoldings of cell membrane. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structure. The outermost, the glycocalyx, followed by the cell wall, and then the plasma membrane. The cell envelope consists of tightly bound three-layered structure, that is, the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs the distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, the scientist Gram, developed by Gram. Those that take up the Gram stain are Gram positive, and the others that do not are called Gram negative bacteria. Okay, so let me take a look here. That based the bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram. Those that take up the Gram stain are Gram positive, and others that do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some. The glycocalyx differs in composition and thickness among the different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called capsule. The cell wall determines the shape of the cell and provide a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is a semi-permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryote. The plasma membrane is semi-permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryote. A special membranous structure is the mesosome. A special membranous structure is a mesosome, which is formed by the extensions of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. Formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to the daughter cells. They helps in cell wall formation, DNA replication, and distribution to daughter cell. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. They also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes, like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. Filament, hook, and basal body. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae, besides flagella, pili and fimbriae are also the surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The pili are elongated tubular structures made of a special protein. The fimbriae, the fimbriae are a small bristle-like fibers sprouting out of the cell. The fimbriae are small bristle-like fiber sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Now, moving on, ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanomicron by 20 nm in size and are made of two subunits, 50s and 30s units, 50s and 30s units, which when present together form 70s. 50s and 30s subunit, which when present together form 70s, the prokaryotic ribosomes. Ribosomes are the site of protein synthesis. The several ribosomes attach to a single mRNA and form a chain called polyribosomes or polysome. Polyribosomes are polysomes. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into protein. The ribosomes of a polysome translate the mRNA into protein. Next, inclusion bodies. Inclusion bodies reserve food material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. Reserve food material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. So, inclusion bodies are the reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Like example, phosphate granules, like synophician granules and glycogen granules. Likewise, gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. Eukaryotic cell possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell wall. That means plant cells possess cell wall, plastids, and a large central vacuole which are absent in animal cells. Animal cells do not contain cell wall, plastid, and a large central vacuole. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Plant cells do not have centrioles. Let us now look at individual cell organelles to understand their structure and functions. The first of all, we'll move with the cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. Meanwhile, chemical studies on the cell membrane, especially on human red blood cells, enabled the scientists to deduce the possible structures of the plasma membrane. These studies showed that the cell membrane is composed of lipids that are arranged in a bilayer. Lipids arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outside and the hydrophobic tails towards the inner part. Hydrophobic tails in the inner part, polar head towards the outer sides. This ensures that the non-polar tail of the saturated hydrocarbon is protected from the aqueous environment. This ensures that the non-polar tail of the saturated hydrocarbon is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglyceride. The lipid component of the membrane mainly consists of phosphoglycerides. Later, the biochemical investigation clearly revealed that the cell membrane also possess protein and carbohydrate. The later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipids. In human being, the membrane of the erythrocyte has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral or peripheral. Depending on the ease of extraction, membrane proteins can be classified as integral or peripheral. Peripheral proteins lie on the surface of membrane, while the integral proteins are partially or totally buried in the membrane. Peripheral protein lie on the surface of the membrane. Peripheral protein lie on the surface of the membrane, while the integral proteins partially or totally buried in the membrane. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson. An improved model of the structure of cell membrane was proposed by Singer and Nicholson, widely accepted as fluid mosaic model. Singer and Nicholson fluid mosaic model. According to this, the quasi-fluid nature of lipid enables the lateral movements of proteins within the overall bilayer. According to this, the fluid mosaic model, the quasi-fluid nature of the lipid enables the lateral movement of the protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Selective permeability. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Many molecules can move briefly across the membrane without any requirement of energy. Without any requirement of energy, we call it a passive transport. The neutral absolutes may move across the membrane by the process of simple diffusion. Neutral solutes may move across the membrane by the process of simple diffusion. By the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower. The neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower concentration. Water may also move across this membrane from higher to lower concentration. Movement of water from higher to lower concentration. Movement of water by diffusion is called osmosis. The movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, as the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher concentration. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized, and it is called active transport. One of the example we have sodium, potassium, ATP pump. Next, cell wall. As you may recall, a non-living rigid structure called cell wall forms an outer covering for the plasma membrane of fungi and plants. The cell wall not only gives a shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell to cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made of cellulose. Algae have cell wall made of cellulose, galactans, and mannans, and minerals like calcium carbonate. Minerals like calcium carbonate. While in other plants, it consists of cellulose, hemicellulose, pectin, and proteins. Let's take a look. Algae have cell wall. Algae have cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while the other plants are having cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth. The cell wall of a young plant cell, we call primary wall, is capable of growth, which is gradually diminishes as the cell matures and the secondary wall is formed. The secondary wall is formed on the inner towards the membrane on the inner side of the cell. The middle lamella is a layer mainly of calcium pectate. The middle lamella, is a layer mainly of a calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata, which connect the cytoplasm of a neighboring cell. The cell wall and middle lamellae may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cells. Endomembrane system. Moving on to the endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the functions of the mitochondria, chloroplast, and peroxisome are not coordinated with the above component, these are not considered as a part of endomembrane system. Let me see. Since the functions of mitochondria, chloroplast, and peroxisome, mitochondria, chloroplast, and peroxisome are not coordinated with the above components like endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. That's why mitochondria, chloroplast, and peroxisomes are not coordinated with the above components. These are not considered as a part of the endomembrane system. The endoplasmic reticulum. Electron microscopic studies of the eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm, that is called the endoplasmic reticulum. That hence ER, endoplasmic reticulum, divides the intracellular space into two distinct compartments: luminal inside ER and extraluminal, that is cytoplasm. Luminal and extraluminal. ER divides the intracellular space into two distinct compartments: luminal, which is there inside ER, and extraluminal, that is cytoplasm. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum. The RER, rough endoplasmic reticulum, is frequently observed in the cells actively involved in protein synthesis and secretion. RER, in the cells which are involved that in protein synthesis and secretion, they are extensive and continuous with the outer. They are extensive and continuous with the outer membrane of the nucleus. Rough endoplasmic reticulum are extensive and continuous with the outer membrane of the nucleus. Smooth endoplasmic reticulum is the major site for synthesis of lipids. Smooth endoplasmic reticulum is a major site for the synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. Next, Golgi apparatus. Camillo Golgi, 1898. The first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of zero point 0.5 millimicron to 1.0 millimicron in diameter. These are stacked parallel to each other. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cysts are the forming phase. Distinct convex cysts are the forming phase, and concave trans are the maturing phase. There are two the phases. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cysts are the forming phase. Cis phase or forming phase, trans phase or maturing phase, concave, trans or maturing phase. Cis and trans phases of the organelle are entirely different but interconnected. The cis and trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. The Golgi apparatus principally perform the function of packaging materials to be delivered either to intracellular target or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. The Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Lysosomes. These are membrane-bound vesicular structures. Lysosomes are the membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. These are formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. like hydrolyses, like lipases, proteases, carbohydrates that are optimally active at the acidic pH. These enzymes are capable of digesting carbohydrate, proteins, lipids, and nucleic acid. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, optimally act at the acidic pH. These enzymes are capable of digesting carbohydrate, proteins, lipids, and nucleic acid. Next, vacuoles. The vacuole is a membrane-bound space found in the cytoplasm. Vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. So the vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. The vacuole bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitate the transport of a number of ions and other materials against concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In plants, the tonoplast facilitate the transport of a number of ions in the other materials against the concentration gradient in the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for excretion. In amoeba, the contractile vacuole is important for excretion. In many cells, as in protease, the food vacuoles are formed by engulfing the food particles. Next, mitochondria. Mitochondria singular we call mitochondrion unless specifically stained are not easily visible under the microscope. Mitochondria, unless specifically stained, they're not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size, also, the considerable degree of variability is observed in mitochondria. In terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage shape or cylindrical. Mitochondria typically is a sausage shape or cylindrical, having a diameter of 0.2 to 1 millimicron. Average is 0.5 millimicron, and length is about 1.0 to 4.1 millimicron. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane. Double membrane bound structure, the mitochondria is with the outer membrane and the inner membrane, dividing its lumen distinctly into two aqueous compartments, outer membrane and inner membrane, and dividing its lumen distinctly into two aqueous compartments, that is, outer compartment and the inner compartment. The inner compartment is called matrix. The inner compartment is called matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called cristae. The number of infoldings called cristae. Towards the matrix, the cristae are towards the matrix. The inner membrane forms a number of infoldings called cristae towards the matrix. Crista, singular. The crista, the cristae increases the surface area. The cristae are increasing the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. The two membrane, outer membrane, inner membrane, have their own specific enzymes associated with mitochondrial function. Mitochondria are the sites of aerobic respiration. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses a single circular DNA molecule. A few RNA molecules like ribosomes, 70S ribosomes, and the components required for the synthesis of protein. The matrix also possesses a single circular DNA molecule, a few RNA molecules, and ribosomes, 70S ribosomes, and the components required for the synthesis of protein. The mitochondria divide by fission. And it contains few RNA molecule, ribosomes, and a single circular DNA molecule. Next, plastids. Plastids are found in all plant cells and in euglenoids. They are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. Chloroplast, chromoplast, and leucoplast. The chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. The chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat soluble carotenoid pigments like keratin, xanthophyll, and others are present. In chromoplast, the fat soluble carotenoid pigments like keratin, xanthophylls, and others are present. This gives that part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients. The leucoplasts are the colorless plastids of varied shapes and sizes with the stored nutrients. Example, amyloplast store carbohydrates, that starch. Example, potato. Then elioplast store oils and fats, whereas elyroplast store proteins. Amyloplast store carbohydrates, elioplast store oils, and elyroplast store proteins. The majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. Majority of chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micron and weight 2 to 4 millimicrons. So these are lens-shaped or oval, spherical, discoid, or even ribbon-like organelles having variable length like 5 to 10 millimicron and width 2 to 4 millimicron. Their number varies from 1 per cell of the chlamydomonas or a green algae to 20 to 40 per cell in the mesophyll. The number varies 1 per cell. Like chlamydomonas, a green algae 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplast also double membrane bound. Like mitochondria, the chloroplast also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma. The space limited by the inner membrane of the chloroplast is called stroma. The number of organized flattened membranous sacs are called thylakoids. The number of organized flattened membranous sacs, we say thylakoids. Okay. Then are present in the stroma. In the stroma itself, the thyroids are present. Then thyroids are arranged in stacks like the piles of coins called grana, granum, singular granum. See here. So thylakoids are arranged in stacks like the piles of coins called grana or granum or the intergranal thylakoids or the intergranal thylakoids, we can say. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. Here, these are the stroma lamellae connecting that. In addition, what we say, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids includes a space called a lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrates and proteins. The stroma of the chloroplast contain enzymes. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. So stroma also contains the small double-stranded circular DNA molecules and ribosomes. The chlorophyll pigments are present in the thylakoids. The chlorophyll pigments are present in the thylakoid. The ribosomes or the chloroplasts are smaller than the cytoplasmic ribosome. The ribosomes of the chloroplast are smaller than the cytoplasmic ribosome. The ribosomes of the chloroplast are 70 years. The cytoplasmic ribosomes are 80 years. Moving on, ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palade in 1953. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. They are composed of ribonucleic acid RNA and proteins and they are not surrounded by any membrane. The eukaryotic ribosomes are 80S, while the prokaryotic ribosomes are 70S. Here the S is Swedberg's unit. The stands for the sedimentation coefficient. It indirectly is a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Next, cytoskeleton. An elaborate network of filamentous proteinaceous structures present in the cytoplasm, collectively referred to as the cytoskeleton. An elaborate network of filamentous proteinaceous structures present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Next, cilia and flagella. Cilia, singular cilium, flagella, singular flagellum, are the hair-like outgrowths of the cell membrane. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are the small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Cilia are the small structure which work like oars, causing the movement of either the cell or the surrounding fluid. The flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but they are structurally different from that of eukaryotic flagella. The electron microscope study of cilium or the flagellum show that they are covered with the plasma membrane. Their core, called the axoneme, possess a number of microtubules running parallel to the long axis. The axoneme usually has 9 pairs of doublets. The axoneme usually have 9 pairs of doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of the axonemal microtubules is referred to as 9 plus 2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublet by radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structures called the basal bodies. Both the cilium and flagellum emerge from the centriole-like structures called basal bodies. Next, centrosome and centriole. Centrosome is an organelle usually containing two cylindrical structures called centriole. Centrosome is an organelle usually contain two cylindrical structures called centriole. They are surrounded by amorphous pericentriolar material. Centrioles are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cart wheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. Here, each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also a proteinaceous and called the hub. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with the tubules of the peripheral triplets by radial spokes made of protein. The centriole forms the basal body of cilia or flagella. Centrioles form the basal body of cilia or flagella. And spindle fibers that give rise to the spindle apparatus during cell division in animal cell. The centrioles form the basal body of cilia or flagella and the spindle fiber that give rise to the spindle apparatus during division in animal cells. Next, the nucleus. The nucleus is a cell organelle was first described by Robert Brown as early as 1831. Later, Robert Brown is a scientist to be remembered that nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. Name chromatin by Fleming. The interphase nucleus of a cell when it is not dividing, we call interphase nucleus, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. One or more spherical bodies called nucleoli, singular nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nm, called the perinuclear space. It is called perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which the movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of the organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, like erythrocytes of many mammals and sieve tube cells of vascular plants lack nucleus. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contain nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. The nucleoli are spherical structures. The nucleoli are a spherical structure present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. Nucleolus is not a membrane bound structure. It is site for active ribosomal RNA synthesis. The larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contain DNA and some basic proteins called histones. Chromatin protein contain DNA and some basic proteins called histone. Some non-histone proteins and also RNA. It contains a single human cell has approximately 2-meter long thread of DNA. 2-meter long thread of DNA distributed among its 46, that means 23 pairs of chromosomes. You will study the details of DNA packaging in the form of chromosome in class 12. Every chromosome essentially has a primary constriction or the centromere on the sides of which disc-shaped structures called kinetocores. Disc-shaped structures called kinetocores. Each chromosome essentially has a primary constriction of the centromere on the sides of which disc-shaped structures called kinetocores are present. Based on the position of the centromere, the chromosomes can be classified into 4 types. The metacentric chromosomes has the middle centromere, there in the diagram. See the metacentric chromosomes. The metacentric chromosomes has middle centromere forming two equal arms of the chromosome. Submetacentric chromosome. Submetacentric chromosome are centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. We call them as submetacentric chromosomes. Okay. Next, submetacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosomes, the chromosome has the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Telocentric chromosome, which has a terminal centromere. So, metacentric, submetacentric, acrocentric, telocentric. Sometimes a few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of a small fragment called satellite. This gives a fragment called satellite. So sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives the appearance of small fragment called satellite. Finally, microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. The many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animal cells. So this is just text reading only. There is no explanation here. Only the text reading. For explanation, you have to go for the playlist to watch the videos. This is just a textbook reading for our easy revision. Thank you.